Hi, I'm Connor Lohan, and I'll be teaching you a little bit about proteins, and I'm using Prezi and Screencast-O-Matic. Hope you enjoy. What do you think of when you think of proteins? Do you think of protein shakes? Do you think of the meat? Do you think of what do you think? Well, protein is actually a macromolecule. There are four classes of macromolecules. There's carbs, there's lipids, there's nucleic acids, and there's proteins. And these are all things that are essential to our bodies, and we need to take in our food, in our daily diet, so our body can function. Protein is extremely important. Proteins are a type of polypeptide. A polypeptide is a polymer made of amino acids connected in a specific sequence. As you can see in this diagram, this is a polypeptide. So you have the peptide bonds between carbon and nitrogen. You have the R groups. This is a type of polypeptide. The protein shapes they differ. They can have the function of the protein depends on the shape. And the proteins begin as chains of amino acids, and then the amino acids determine the shape. Amino acids, as I said before, make up the proteins, and they determine the function of the proteins. And as you can see, in this chart, there's 20 amino acids. You see the names here, glycine. There's the formula, abbreviations. There's 20 of these, but only 9 of them are essential. And these are the ones that are in green here. So valine, all the way to valine to this one. So yeah, the order of the amino acids determines the shape and function, and there are four levels of protein binding. There's the primary, secondary, tertiary, and quaternary. The proteins, their functions, they differ. Proteins can be used for structural support of cells, storage, transport, intercellular signaling, movement, immunity, and enzymes. The primary structure is determined by genetic information, and a slight change in the structure can affect shape. H bonds cause a secondary structure to spiral or fold, and they determine structural properties. And the tertiary structure controls the interactions between the R groups. The quaternary structure has 2 or more subunits. That's just a little thing about the levels. There are a few things that affect the protein shape, and therefore the function and how well it can function. So the environment, you know, that whatever happens in the environment that can affect it. Also, the misfolded proteins cause the protein not to work, and misfolded proteins. Examples of those are Alzheimer's disease, BSE, and Parkinson's disease. Denaturation also affects the function of the protein and how well it works. And as you can see here, there's fine alpha helix, but after denaturation, it has no specific shape and can't work. Denaturation can be caused by alterations in pH, salt concentration, temperature, and other factors. Alright, well, I hope you learned a little bit about proteins and amino acids. Thank you very much.